app. And I was going to first introduce myself because I know it's my first time up on stage. Fred mentioned me this morning. My name is Natasha Christie Holmes. I'm the Director of Partnerships and Strategy for EPIC, the Emerging and Pandemic Infections Consortium, and we're very happy to be partnering again on this symposium for the fourth year in a row. Very excited to be here again. We're very excited always to coordinate the engagement of trainees with the symposium and specifically help coordinate abstract submission and selection for our poster session that'll take place today between 5:30 and 7:30 at the end of sessions. But we're also, through gathering those abstracts, we were very excited to be able to go through a selection process to have 3 of our talented researchers take the podium at different times during the symposium. So right now I'm going to introduce our first selected speaker, Mahmood Gohari, who is a postdoctoral fellow working at Public Health Ontario, and he'll be presenting on predicting antimicrobial resistance in Shigella using machine learning and whole genome sequencing.
Good morning, everyone, and thank you very much, Natasha, for the introduction. Let me see. Just. All right. Perfect. All right, so today I'm presenting our work on predicting antimicrobial resistant, drug-specific antimicrobial resistance in Shigella. So using whole genome sequencing and machine learning, we've built an end-to-end pipeline that takes the genome, raw genomic reads and can flag which isolates are likely resistant. So today the story is about using whole genome sequencing and machine learning methods to predict antimicrobial resistance in Shigella. Before I get into modeling, let me talk very briefly why Shigella matters. So Shigella species are one of the leading causes of bacterial gastroenteritis worldwide. Every year we see millions of cases and estimated 100,000 deaths globally. So this is not a rare infection, it's a real public health challenge. Among the Shigella species, Sonne is the most common species, while dysentery is associated with the highest mortality. Now the challenge with Shigella is not how prevalent it is. The real challenge is how resistant it has become. Around the world, we are seeing extensively drug-resistant strains emerging, and recently WHO announced Shigella as a priority pathogen for AMR surveillance. Here in Ontario, about half of the Shigella cases were multi-drug resistant, and we have already seen XDR cases locally. So from both a public health perspective and clinical perspective, Shigella is a real challenge. As we know, the way we determine whether a strain is resistant is through AST. This is a traditional and gold standard method of detecting resistant samples. But the challenge with AST is that it takes time. You need to culture the organism, expose it to multiple antibiotics, and wait to grow. That means the process can be slow and resource intensive. To help address some of the limitations of AST, traditional AST, whole genome sequencing can be used and can address some of the limitations. We can turn to whole genome sequencing. With WGS, we are able to look directly at the organism's DNA and identify the sequences and identify all of the markers that are associated with resistance. Here's a quick overview of how samples move through Public Health Ontario. So we need to do this machine learning methods, we need to have both AST results and also genomic sequencing data. So here is the flowchart, and here's how data currently is produced at Public Health Ontario, and I have access to this data to be able to test my model of that. So just a very quick overview. When a clinical sample arrive at Public Health Ontario, and they got the sample positive, Shigella positive, they can do the species identification and typing, and then at the same time, all the isolates are sent to National Microbiology Lab for sequencing. Once it's done, after sequencing, the results sent us back to Public Health of Ontario, and then we have both data sets, phenotypic data and also sequencing data. And for this research, I have combined these two data sets to have 1,548 samples with both AST results and also sequencing. So this is the foundation of what I'm going to do with machine learning and testing our model. So this brings us to the main goal of our research. The idea was to take whole genome sequencing data and use it to detect AMR in Shigella. More specifically, we wanted to build a model that could tell us which isolates are resistant and which genomic features derive that resistance. To do that, we use machine learning methods that can go through the genome and highlight the markers most strongly associated with resistance. Here's the pipeline we built in to turn raw genomic data into a structured data set for machine learning. Very briefly, after running quality control at somewhere here, after running these quality controls, we extracted known genome information for each isolate and then generated the k-mers. This result is in a very large data set. So building this k-mers matrix of, it's a huge matrix of data. When we have this, it's kind of the millions of k-mers per isolate, and this k-mers table becomes the input for our machine learning method. So what we put into the machine learning is a data set of k-mers. We used some feature engineering methods to reduce the dimensionality of data set. We then split data set into training and test data. For any of machine learning methods, we need to train the model and then test it on a separate data set. So we use 70% of our data for training and 30% of our data for testing the model, how our model, our predictions performs on data sets. We evaluated several machine learning methods, including random forest, XGBoost, logistic regression, and support vector machines, different methods to find and predict resistance of our isolates. I'm going back. And after training, when the training is done, we just test the performance of our model on test data. Just I need to mention that this training is an iterative process. We do it repeatedly to get the best performance, to get the best, we call it hyperparameters. We tune them. We repeat this process multiple times to have the best predictions. It's not just one time running a model. After evaluating the model, there are many different ways to evaluate the performance of our prediction. I will mention it in a couple of slides later. So here's the phenotypic results for the 9 antibiotics in our data set. Today I'm focusing only on ciprofloxacin, one of the key treatments for Shigella, but we have information for other antibiotics as well. So ciprofloxacin in our data shows that for almost 47% of them are resistant, 40% resistant, 47% susceptible, and 30% of them are classified as intermediate. Why we choose this ciprofloxacin? Maybe just very briefly that because we already know the key genes and mutations that causes resistance to this drug. So this would be a really good case test for us to evaluate our predictions because we know all of the background of that. So this is a good test case for our model. So for each isolate, as I mentioned earlier, in the workflow, we created k-mers and the k-mers here is 21 base pair of k-mers, meaning that we created a data set of 21 k-mers, given the size of the Shigella genome, which is something between 40.5 to 5.5 million. So you can imagine that there would be thousands of these k-mers is created, and it's, again, would be a very huge matrix to handle, to input to the, our model. So this is essentially a large matrix showing that, so what we show in this matrix is that for each of these isolate, if a specific k-mer is presented or not. It's a binary present, or we call it presence-absence matrix, so we have this information. Again, it's a big one. And this is exactly where machine learning becomes useful, because statistical methods, any other traditional methods cannot handle this large data set and this level of complexity. We need to go with the machine learning methods to handle this complexity of data. So as I mentioned, we have used different methods, machine learning methods. One of them is just random forest method, just very, very briefly giving you an idea how these models work. So random forest is an expansion of another model. We call it decision tree. By decision tree, it's a very simple method of splitting data by answering a yes and no question. So each of trees split data and find the list of important k-mers by saying that if this is important or not. I'm going a lot of things back of the method, but the idea is very simple, right? So just finding a list of important k-mers by using a decision tree. But this decision tree, we cannot rely on only one tree. It's better to expand it and use random forest to create hundreds of these forests, these decision trees. And then how it works, the decision tree, at the end of the running the random forest, they can vote which list of k-mers are the most important ones. It's like a very democratic way of choosing the best one. It's a true machine learning democracy. It's happening here with random forest, right? It's all based on the voting. So here is the result of random forest. It's an output of random forest. It's showing some key performance of our model. We use 70% of data again for training, and this is the result on test data, the data that it hasn't been seen by models. So we can imagine that this would be kind of a new data set that we are implementing our data. There are many different metrics to measure the performance, but I wanted to highlight here two of them, precision and recall. Our model estimated precision as 95%, as you can see here, and recall as 90%. So what does it mean, the precision, when we say that it's 90%? It means that when our model predicts an isolate as a resistant, in 95% of cases, it's true, it's correct. So the correct predictions, right? What the recall means here, it means that when our the predict or recall is 90%, our model was able to correctly identify 90% of true resistant isolates. Those are two different things, right? So it means that missing a resistant sample is only 10% by our predictions here, because we are able to pick 90% of resistant samples, true resistant samples. And here's another way of showing the results. So we call it this confusion matrix, this plot. Compare the model's predictions with the phenotypic AST results. So the blue square shows where the model and the AST results match. So we can see that there are lots of match results here. But of course, like any other model, any other predictions, there are some inconsistency. No model is perfect, right? So we have some misclassified here. There are 13 samples here, resistant sample misclassified as susceptible here. So it's 13% here resistant by AST, but predicted as susceptible. And you can see another misclassified piece, 4 cases or isolate which were susceptible, but our model predicted as resistant. And this is the way that we can find that where our model need to be improved and how we can improve our model, where exactly which cases we can identify, which cases we have misclassified them, and then going through and finding the reason for that misclassification. But this confusion matrix gives us a clear picture of both maybe the strength of our model and the weakness of our model, because it shows both match and mismatch cases here. So far, after generating all those millions of k-mers and checking how well the model performs based on these measures, the next thing we wanted to do, and is very important, is making sure the model is actually learning real biology. It's not picking random k-mers, right? Not random guessing. This is important because we can, have by any mathematical method, statistical method, machine learning method, we can pick something, right? By random, by probability, that kind of thing. But we wanted to look at how interpretable are those identified k-mers. So, in other words, when the model says certain k-mers are important, we wanted to ask, do these actually map to known resistance mechanisms or not? Do they make sense biologically? And here's a list of some identified k-mers as important k-mers for, again, just only CPRO resistance. So when we map these k-mers back to the genome, we found that all of those identified important k-mers map to GIRC and PAR-C and the related mutations. So this is a well-known marker for CPRO resistance, and it's assuring us that our model is picking right k-mers, right genes. It's not just, again, random guessing and based on all pure mathematical number picking their values or k-mers. So to wrap up, we developed a machine learning pipeline that can reliably separate CPRO-resistant Shigella isolates from susceptible ones. We can now go from raw sequencing here to making decision if an isolate is resistant or susceptible by receiving a new isolate, sequencing, and then putting into our model. We are able to make a decision to see if it's resistant or not. I should say that this is the first phase of our work. The ultimate goal of our work is creating a tool to be able to use it for making these predictions in function, in day-to-day use in labs or anywhere we need to identify resistant samples. To reach out that goal, we need to first evaluate the model. So this is, again, the first piece of that. There are a lot of things need to be done yet, so long way to go. The next step for us is to just validating our model on external data, also evaluating on other antibiotics. We have it just for CPRO. We need to work on other antibiotics. We need to do more machine learning works to reduce the dimensionality. Dimensionality is very important here to future refinement. And lastly, but very important part is biological interpretation. Again, going back to find that if all of these k-mers, all of these genes identified by this machine learning method are interpretable, are making sense by biology. That's the thing that we did for CPRO. We need to do it for other drugs as well. And yes, that's it. So a big thank you to my supervisor and co-supervisor and the Public Health Ontario team, which are here, for their invaluable support and contribution. And thank you for listening. Happy to take some questions.